My name is Julijana. I am truly honored to be here and serve as a moderator for the last Young Scientist presentation session called Physical Sciences, All Things Small. So as you have heard, I'm the first speaker. So I'm research scientist at Skolkovo Institute of Science and Technology in Moscow, in Russia. And today I stand before you to unravel some mysteries of diatom algae, highlighting their extraordinary optical and mechanical properties, as well as the power these properties hold in terms of the science and technology. I wanted to start this presentation by showing you this mesmerizing video, unveiling the intricacy hidden in this single diatom cell consisting of a delicate lattice of pores inside which those green vibrant chloroplasts are located and serve as energy powerhouses, while lipid droplets stand ready to fuel the diatom's journey through life's ebbs and flows. Actually, this video serves as a reminder that there is a world of complexity hidden even in the tiniest of beings on our Earth. But what are diatoms? Diatoms are an extremely diverse class of microorganisms with exoskeletons composed of hydrated, highly porous silica called frustules. They attract generations of scientists of different specialties owing to their fascinating structure, abundance, functionality, and unique properties. But still, why should we care about them? Because their ecological significance is staggering. Not only they anchor marine food webs, but they also have an outsized atmospheric impact. It is roughly estimated that 1/4 of the air we breathe is created by diatoms. And as a result of photosynthesis, they produce biomass rich in proteins, lipids, and other valuable molecules, and at the same time represent a source of nanostructured amorphous silica with huge potential for future applications in modern science and technology, as well as in industrial applications. So our journey here begins with a quest to answer to the question of Charles Darwin. Were these stunning structures meant to be admired only under the microscope, or did nature craft them for a more profound revelation? So as Darwin hinted, we can look into the world of diatoms through the lens of a microscope and consider, for example, such a multi-layered structure of centric diatom species. So the diatom frustule actually consists of these two valves that are overlapping each other in a petri dish-like arrangement and a girdle band located between them. And each valve comprises 3 interlinked silica layers with different pore sizes, thickness, and pore distributions. And such morphology actually bears resemblance to the membranes in MEMS microphones, prompting our decision to study the relationship between this hierarchical structure, multi-level structure, and mechanical properties at micro and nanoscales using atomic force microscopy and an in situ nanoindentation inside the column of scanning electron microscope. Atomic force microscopy helped us to obtain these detailed Young modulus maps of cleaned inner surface of diatom, clean outer surface, which means that they don't have any organic components inside or outside of the shell. We have just like silica shell, and to compare them with the wet living diatom cell. And actually, the results that we obtained were in some manner logical. Why? Because the highest values were obtained from this wet diatom cell, around 25 gigapascals. It is quite expected considering the support from the organic material within. But why do we see differences between these cleaned inner and outer surfaces? Well, it turns out that the stronger mechanical response from the cleaned inner plate can be explained by the fact that it serves as a basic framework for building other porous layers on during the last steps of frustule formation. Since actually AFM gives us detailed information about material but under local indentation, we performed in situ nano indentation inside the column of a microscope. What you see here is actually the diatom frustule undergoing cyclic loading. We see that it tends to oscillate even after this initial rupture at the end. And based on such behavior, we calculated the compliance as the inverse of stiffness and obtained values which actually shows us a good potential for future MEMS applications of diatoms. And this actually relationship between structure and mechanical properties has a direct impact on resonance frequencies of diatoms. So what we did is to first perform numerical calculations of the eigenfrequencies using COMSOL multiphysics, and then to compare them with experimental values. And for the first time, we experimentally confirmed these numerically predicted calculations using the optical system of atomic force microscope. So this correlation between experimentally obtained and numerically predicted values is highlighted here by these color bars. And precise measurements of resonance frequencies is very important for the optimal design of next generations of these MEMS membranes. Now I would like to switch to the optical properties to see how this light interacts with such periodic porous structure. And for that purpose, we performed simulations of light intensity distribution inside the diatom shell on this 3D numerical model using Fourier model method. So we obtained the next results. So you can see that if we are looking at this vertical cross-section of the diatom frustule, that there is an interference pattern with those bright spots as well as dark regions. And the intricacies of these light interference patterns leads to localized areas of high photon concentrations, and this phenomena actually could facilitate the optimal light utilization by chromophores, namely chloroplasts, which are situated within these high-intensity regions. And therefore, we can expect that this can facilitate further energy absorption as well as photosynthetic efficiency. Given that diatoms usually thrive at depths around 10 to 50 meters below the water surface, where there is enough light, let's take a quick look at the solar spectra depending on the water depth. We can see that beyond these 10 meters, actually the red visible light diminishes, so we only have like blue light left. And what is interesting is that if we take a look at this absorption spectra of living diatom cell, actually the absorption maximum of chlorophyll A, which is the main chromophore in diatoms, is exactly in this region. But what about reflection? What about transmission depending on the angle of incidence? So we see that the reflection actually is merely observable at these angles which are close to the normal incidence angles, which aligns with the fact that there is a small contrast between refractive indices of media, which is water, and material, which is silica. Transmission is minimal at this region of wavelengths 500 to 400 nanometers, and while extinction actually has the opposite values. Extinction is maximum in this region, and it decreases with increasing wavelengths. So understanding optical characteristics is very important for design, bio-inspired design aimed at enhancing photosynthetic efficiency for different applications. For example, even for design of bio-inspired photovoltaic cells or advanced photonic materials. And to sum up, the study of diatoms with its blend of optical and mechanical properties propels us towards a future where these microscopic wonders serve as catalysts for innovation. Their sustainable design principles, mechanical resilience, as well as this magical prowess at manipulating light at different scales can help us to develop materials for different applications, electromechanical, photonic, liquid, and many others. Before I conclude this talk, I would just like to express my deepest gratitude to all colleagues who were part of this study, RSF grant and Clover project, for financial support, and thank you for joining me on this journey into the atom world. Thank you.